Hi, welcome to this lecture on environmental adaptation of animals. On these few videos, I will have a short review on this fashionable biology word omics. And we will start with genomics that reminds me on genes, but because it's omics with the plural is S, it has to have more than one gene. So it has a lot of genes. And let's have an example. This is Antarctic ice fish living in the ocean beside the Antarctica. The temperature of this environment is very constant. It's throughout the year minus 1.5 degrees of Celsius, and you can see that this ice fish has very peculiar color. It's very pale. And the reason why it's pale is that actually sometimes not only the small changes in enzyme affinity are enough. So in these two test tubes, there are blood. On the right hand side, there is a regular blood sample. And in the left hand side, there is a regular blood sample of this Antarctic ice fish. No red. When looking at the gills, you can see also that the gills are very pale. And the reason why it's so pale, it's because there is no hemoglobin. And well, you all know that hemoglobin is needed for transporting oxygen throughout our circulation. So it has very limited oxygen carrying capacity in the Antarctic ice fish. It can carry only 10% of the oxygen to compare the close related red-blooded species. And well, they compensate this with large changes in the cardiovascular system, and one of these changes is that they have large blood volume and high flow rate. So then they can have a fast circulating blood without a high blood pressure. And when it's circulating fast, it's getting icy very difficultly. But how this hemoglobin can be lost? Well, we have genes and pseudogenes. So we have genes that are coding proteins, and then we have genes that are not able to code proteins, and these are pseudogenes. So in vertebrates, the hemoglobin contain alpha and beta globin subunit. They both contain 3 exons and then 2 introns between them. And these genes are very ancient. They can be found in bacteria and yeast, etc. And in mammals, like me and you, we have these beta globin and alpha globin in 2 different chromosomes. But in teleost fish, these genes are quite close each other in the same chromosome. And in most ice fish, they don't have beta globin gene at all. And this alpha globin, it only have the exon number 3, making this gene completely non-functional. And surprise, surprise, that's why they don't have hemoglobin at all. So how does this can be evolved? Well, first of all, the good thing is that, we should perhaps first make some kind of family tree that what kind of fish these ice fish are. And the family trees can be done with the genomics by comparing mitochondrial DNA. Mitochondrial DNA, beside this DNA in the nucleus, there can always be some spontaneous mutations throughout the evolution. But in the mitochondria, they can change the nucleotides by mutations a little bit easier. So that's why we can track the fast evolution quite nicely with this mitochondrial DNA. And the good point also is that mitochondrial DNA does nothing to do with hemoglobin. So which means that, if they are very close to each other, then there's a short time from the common ancestor. But if they have a lot of mutations, a lot of differences in this mitochondrial DNA, then there is more time from the common ancestor. And then when comparing different species, we can have some kind of family tree based on these genetic differences. And we can place these species to group close relation or more distant cousins. Like over here, you can see that one branch with containing these on the lowest part, they are quite far from these two different red-blooded fishes. But then, when adding these ice fish, we can see that they have similarities with the mitochondrial DNA, and they are coming from the same branch. And that means that, okay, they have a single ancestor, which was the first ice fish in the evolution. And when comparing this with the loss of globin coding genes, the globin genes has been lost in all these ice fish. And most probably it has happened only once in the history of life. So the most logical spot of the loss of hemoglobin has happened just when the first ice fish were evolving. And one of the reasons is that, okay, not only that they have lost this globin, but it's also they have all lost beta globin, and the alpha globin is non-functional fragment. So most probably this kind of thing doesn't happen every day, but it has happened only once in these fish. Well, hemoglobin is not the only globin in our body, and not only that is interacting or binding oxygen. We also have myoglobin in our muscles. And these, in several ice fish, they also lack the myoglobin. But as you can see, that in all these with gray, they don't have myoglobin at all. But it has happened several times before because they are in several branches in this tree of evolution. And how this myoglobin is then affecting? Well, there's two hearts of ice fish and then one heart of red-blooded fish on the right. As you can see that in both cases, the heart is very large to compare these red-blooded fish. But you can see that the ice fish without the myoglobin, it's also very pale. Okay, so we can have animals without hemoglobin and without myoglobin, but also only in very specific places on this planet. And the problem actually is that the Antarctic Ocean, the temperature is minus 1.5 degrees of Celsius, so it's below zero. And fish, actually, although they are living in the ocean which contains a lot of salt, they have quite low salt content. So they have more or less similar salt concentration, sodium concentration, chloride concentration in their blood as you or me. So that's why these fish, their body can easily freeze, although the surrounding water is still liquid. And in later videos, we will describe more on this antifreezing, but actually we can reduce the freezing point of liquid by adding actually any kind of substance in the solution. And all the animals that are, all the fish that are living in this Antarctic Ocean, they have these antifreeze glycoproteins. So it doesn't anymore matter if they have the ice fish or these red-blooded fish. There has been appearance of the genes coding for antifreeze glycoproteins. So that's most probably has happened even prior the evolved of the first ice fish. And of course, the interesting is that, okay, why can we have some antifreezing proteins in these fish? Well, actually, they just modulated enzymes that were released by pancreas. So again, not to evolve completely new protein, but to change the properties of the old ones. So this is just one example of using genomics. Well, because the problem in when studying environmental adaptation can be that when we notice that, okay, animal can tolerate, for example, cold temperature, it's very difficult to find out that, okay, which gene or which protein we should test. That, okay, and if we fail for one time, can we just take the second protein to see that, okay, was that affecting, or the third one, etc. And that would take a lot of time and a lot of effort. And in genomics, we can then check the whole genetic material. And the classical example of genomics is the Human Genome Project, where we know each nucleotide in the human genome and to compare that, okay, what has happened or how they function. And after the Human Genome Project, this has been expanded on several other animals also. But actually, genomic study can be done by resolving the origin of the genes, for example. So we don't have to always compare all the genes or thousands of genes for this genomics. But when we are studying only single gene in a single organ, that's genetics, not genomics. Well, genomics has actually, why this kind of result or research has been done, it has two goals. The genes and for the genes and genomes, so to elucidate the evolution. Because it's, of course, we are interested on how, for example, antifreezing glycoproteins have been emerged in the animals, but most probably this latter one is the more important reason to elucidate the current functioning. And examples on this has been done a lot of in human medicine, for example, adult type diabetes. So when testing the whole human genome to see that, okay, which genes are evolved in the diabetes, the understanding has been expanded. So it's not only anymore dealing with the sugar metabolism, but it's doing a lot in different kind of organs, etc. And when we are comparing genes, we can find genes with structural similarities. So meaning that, okay, they have the same nucleotides in the same order. So they are homologous genes, and they form gene families. So usually when we have, for example, unidentified, completely new protein in animal, we just compare that, okay, have anyone published anything that is looking the same kind of gene? And if they have two different genes that have structural similarities, most probably these proteins have functional similarities. But not always, because we can have enzymes that were usually released for digestion, and now they were used for antifreezing. Well, for genomics, the most important tool for this kind of research is the computer. And quite often, this can be done with any kind of computer by using some web pages, like NCPA, from where you can find any kind of protein and what kind of amino acid does it contain, etc. But then also you can find that, okay, you can make also the blast. And when you enter on the blast, you will get this kind of weave. You can compare any nucleotide to all the nucleotides that have ever been published, or any kind of protein sequence to all the protein sequences anyone has published. Or then you can blast the whole genomics of certain organism. And when making a protein blast, you can either add the completely amino acid sequence one by one, or then, for example, in this case, I have here human lactate dehydrogenase. I can just copy this gene bank number and just mark the blast. And this will then take a little bit time when it's trying to find out structures or sequences that are similar, and in the end it will look like this. So, okay, seeing that, okay, there is this lactate dehydrogenase that is in some kind of Rosman superfamily, and it has find several sequences that are completely identical, and then comparing to each other. And one of the interesting things is, okay, there is this distant tree of result that will lead to this kind of picture. And over here you have the lactate dehydrogenase of human, and then some other monkeys, then some bats or carnivores, etc., dolphins. So how far from each other these proteins that are doing the same thing in different organisms, how far they are from each other. So it's not so difficult. But when having the whole genome, we can find actually quite interesting research. And for example, these gene families in sea urchin. So sea urchin, one of the model animals in the development biology, for example, when they get the whole genome sequenced, they found out that there are no genes for JAP functions. So they don't have JAP functions in these organs at all. And there are no genes for epinephrine or melatonin, but they have genes for other neurotransmitters. So most probably these other neurotransmitters have emerged first, and then these epinephrine and melatonin a little bit later in the evolution. And these animals, they have a lot of genes coding elements in the immune system, and plenty of genes related on detoxification. So they live in swallow waters, a lot of microbes, etc., and pathogens, and they get to fight against these pathogens constantly. And then, when comparing it, we can also see that, okay, there is no there are differences in the skeletal mineralization from vertebrates. Well, they don't have bones. And also there are many genes that are thought to exist only in vertebrates. So sometimes when having the whole sequence of animals resolved and sequenced, we can find that, okay, there are some answers that we didn't even have a question for it. Okay, and that's why this genomics has actually changed the way that we think that, okay, how these studies should be done. Well, in the old school method, we know that, okay, the genes are affecting on the proteins that exist in the tissue, and they are affecting on the function of the tissue, and then they coordinate the animal function. But still, when we are doing the research, we see that, okay, this animal is functioning like this, so it's running fast. Okay, which kind of tissues are affecting on the running? well, the muscles. And then, okay, which kind of proteins are affecting on these muscles to contract? And then, okay, what kind of genes are evolved in these contracting proteins? So it's top-down order of study. But with this genomics, we actually now know that what kind of genes exist, are present, or what are transcripted. So what kind of proteins exist in these tissues? And then, when we know the proteins, how should this tissue most probably function? And then, when knowing the function of this tissue, it can affect that, okay, how this animal is functioning. So that's why we can have questions in two different ways. We can have top-down order or bottom-up. And thank you.